Hello, this is Mushfiq Hassan, a member of Boal Group. We are the students of 15 batch Fisheries and Marine Science, Noakhali Science and Technology University. Fecundity of fish is the number of total eggs that are laid by a fish in a spawning season. We used a mirror curve to estimate the fecundity. This content is a part of our lab work on fish fecundity estimation. Fecundity can be estimated by gravimetric method, volumetric method, on-bear method, or automatic egg counter. In this video, we used the gravimetric method and the volumetric method to estimate the fecundity.
Before starting this lab work, we must follow some precautions. Those are, we must wear lab coat, eating, drinking, smoking, medication and mouth pipetting should be forbidden. Good hygiene should be maintained. Fish should be collected during peak spawning season. Each gonad portion should be weighted carefully. It should be counted accurately. Instruments used in this lab work are weight machine, tray, dissection tools, petri dish, scale, measuring cylinder, pipette and gloves. Gravimetric method. At first, we weighted the fish and the fish was 900 grams. Then we put it on a tray and took the measure of total length, which was 37.5 centimeter. Standard length 30 centimeter. Fork length 32.5 centimeter. After that, we faced up the ventral side and carefully cut the ventral region from the genital bore to near the operculus side. Then we collected the gonad from the fish and removed its offal. We cleaned the gonad with blotting paper and removed the external membrane. Then we weighted the gonad and found the result 109.6 grams and separated the anterior and posterior lobes. Firstly, we weighted the both lobes. Anterior lobe 59.52 grams. And the posterior lobe is 49.01 grams. Then we cut each of the lobes into 3 portions: anterior, middle and posterior. And then randomly took 3 subsamples from each portion. We denoted the subsamples of both lobes as F1, F2, F3, F4, F5 and F6. From the anterior lobe, we weighted the subsamples. Here, the F1 subsample weighted 121 milligram. F2 subsample weighted 75 milligram. And F3 subsample weighted 286 milligram. Similarly, from the posterior lobe, we weighted the subsamples as F4 subsample
which weighted 330 milligram. F5 subsample weighted 156 milligram. And F6 subsample weighted 157 milligram. Then we counted the number of eggs from each subsample. In F1 we found 52 eggs. In F2 we found 49 eggs. In F3 we found 193 eggs. In F4 we found 247 eggs. In F5 we found 112 eggs. And in F6 we found 99 eggs. Okay, let's jump into the calculation. By using this data, we estimated the fecundity of the mirror carp by using the equation you can see in the slide. Here, the fecundity is equal to number of the eggs in the subsample into gonad weight divided by the weight of the subsample.
Now let's move into the calculation of gravimetric method. For this, at first we estimated the total weight of the ovary. Then we make two portions of this ovary, anterior gonad and posterior gonad. And we have estimated the weight of these two portions. Then for the anterior gonad, we make three subsamples, F1, F2 and F3. At first we estimated the F1 subsample weight, then we counted the number of eggs in this subsample. For the fecundity of F1, we use the formula number of eggs in the subsample multiplied by the total gonad weight and divided by the weight of the subsample. In this way, we have calculated the fecundity 2 and fecundity 3. For anterior part fecundity, we sum up the F1, F2 and F3 and then divided by. We have followed exactly the same procedure for the posterior loop fecundity. For this, we make three subsamples, which is denoted as F4, F5 and F6, and calculated the fecundity of this subsample, and then make a sum up of these three subsamples and divided by 3. For finally the fecundity, the anterior portion fecundity and the posterior portion fecundity is sum up and divided by 2. That's the procedure of the gravimetric method.
Volumetric method. At first, we took 300 ml water in a measuring cylinder. We took whole gonad in a tray. Then we carefully submerged the gonad into the measuring cylinder that contained 300 ml water. We stir the solution for proper mixing. The volume of the solution was raised up to 400 ml, which is the total volume containing all the eggs. Then we cleaned the pipette, rinsing with water. We took two subsamples from the solution by using pipette, where the volume of each subsample was 5 ml. After taking first subsample from the solution, we put it in a petri dish, which is the subsample 1. Similarly, we had taken another subsample, which is subsample 2. We counted the number of eggs in each subsample, subsample 1 and subsample 2. where we found the number of eggs in subsample 1 744, which is denoted by N1, and the number of eggs in subsample 2 829, which is denoted by N2. By using this data, we estimated the fecundity of fish. You can see the equation in the slide. Here, fecundity F is equal to N into capital V divided by small v and represents number of eggs in the subsample. Capital V denoted as a volume which contain all the total egg. Small v represent volume of the subsample.
Now let's move into the calculation of volumetric method. For this we counted the number of egg in each subsample, which is denoted as S1 and S2, where we found the number of egg in subsample 1 is 744 and the number of egg in subsample 2 is 829. By using this data, we estimated the fecundity of fish. You can see the equation in the slide here, where N is denoted as number of eggs in the subsample, capital V is denoted as the volume which contain all total egg, and small v is denoted as the volume of the subsample. For sample 1, the fecundity is estimated by the formula N1 into capital B divided by small v, and for the subsample 2, fecundity is estimated by the formula F2 is equal to N2 into capital V divided by small v, and finally fecundity F is equal to sum up of F1 and F2 divided by 2. This is the process of whole volumetric method.